Okay, glad to have you guys. And as I go through this presentation, keep in mind that the way it was developed was came to RPI about a year and a half ago and started kind of digging through all the information they had and collecting the information that was coming in. So some of it is not really a full site from beginning to end, and I'm not really doing a site presentation, I'm doing data presentation. That doesn't mean that we don't intend on filling some of this out more so than I'm presenting to you today. But those of you who work for companies, you know how it is. Sometimes you're in the middle of collecting great stuff and they say, that's good enough, the site's closed, and there you go, that's where it stops. But let's go to the first. But what I want to show you from this picture is that microbes grow more abundant on activated carbon than they do on most materials. This is not our work. You'll see the published citation down below the picture. On the left-hand side of the screen, you see microbes growing on clay material. And I have a little red circle around the microbe so you'll know what it is. Versus on the right, microbes growing on activated carbon. And what I want you to take from this picture, this is just one example of probably more than a dozen different pictures you can find in the literature and probably over 100 citations to microbes growing rich on activated carbon relative to other materials. Now, this is a picture of microbes growing on activated carbon and building up a biofilm. So there are two stains here. The green stain are live cells, the red stain are dead cells. And you see this shape is held together by the biofilm structure. Now, this picture and the previous picture are both laboratory studies. What you're interested in probably is, well, what happens in the field. So here I have a picture on the left. You see a 2.5 or 2.25 inch bore log that's been sliced vertically, and you'll see a small carbon seam through the bore log. And I'm using this bore log as an example. This isn't the exact same bore log I did the picture on the right from, but it was similar. So I took a bore log that had carbon in it, and on the right is a picture from a confocal microscope set for intensity, because if you just looked at the microbes, it didn't really kind of do the picture justice. The green vertical lines are intensity peaks from the microbes. And what you see is the carbon seam has a lot of intensity against it, where the aquifer material is largely dark. Now, if you look carefully in the aquifer material, like right around in here, you can see some small peaks. So it's not like it's completely bereft of microbial life. It's not. It's just not as rich as one would hope. So those would be right there as an example. So the goal of showing you this is to try to give you a sense. It's not proof by any means, but it gives you a sense that microbes like growing on activated carbon. They grow abundant on activated carbon. Next question would be, yeah, sure, microbes like growing on activated carbon. You can grow them in the lab and you can show me this from a core drawn from the ground. That's cool. But when you actually inject an activated carbon remediation product, like the BOS 200, do you get distribution? So these are core logs from a site, and you can see the carbon distribution in the core log. You look on the left and you can see it there at about 1.75 inches, a nice heavy seam. And then as you look on the right, you see a number of featherings, say at about 25 inches to around 29 inches. And I just show you this to kind of give you a sense of what the material that we've worked in looks like. Now, this was a site in which we pulled 40 cores from the site. And we looked at each of these cores over 2-foot intervals and logged the sightings of carbon. And it wasn't just RPI folks who looked at this. It was also a group of state folks who looked at this. And what we saw was at the 2-foot interval, we had 53 samples out of 273 at 6 to 8 feet. So that's right around the mean, right? Take 273 divided by 5. Where are you, right? And you see that we danced on either side of that mean reasonably well. And so I think that this kind of gives you a sense of what the distribution looks like. The 36 is a little low, the 65 is perhaps a little high. Why is this important? Because most of your contaminant is absorbed to the soil phase. I give you this example of total volatile hydrocarbon mass and say, okay, 82% is absorbed to the soil phase. Now, that's going to vary depending on soil types, but it is always the majority. So you have to have an adequate number of soil samples to actually characterize a site. And what you do to impact what is bound to that soil really determines whether that site is actually clean or not. So here's a site. This actually comes from Denmark. And what you're looking at is your total volatile petroleum hydrocarbons concentration across the top. Then vertically, you're looking at depth below ground surface. In 2012, samples were taken and they followed the blue line. And you can see the peak underneath the blue triangle of concentration. So you got about 8,000 milligrams a kilogram total volatile petroleum hydrocarbons at that time in 2012. Back in 2015, they resampled and you follow the red line. Now, obviously, and I was showing someone this and they said, well, you can never recollect the same sample, so I don't believe it. And I thought that was kind of a fair statement. You can't really collect the same sample. You can only collect one close to it. Nevertheless, notice how similar the patterning is in 2015 to 2012. Then we came back in 2017 and collected soil cores again, and you get the green line, clearly showing that for this sampling, the concentration bound to the soil was decreasing post BOS 200 injection over time. But I guess one could look at this and say, well, it's just one sample. Can you show me something more? Can you show me this in lots of samples? So again, let me remind you what kind of material we're going to be working in the next picture, because this is from the site in which the following data was collected. So here is the naphthalene data from an aquifer 30 months post BOS 200 injection. And if you look at the graph on your left, this is called a quantile graph. And what's going on with this graph is 0 is essentially your mean. So if you look down across the x-axis, 0 is the mean, a 1 is one standard deviation above the mean, as -1 is one standard deviation below. So if you look at the graph, you will see that our initial concentration prior to BOS 200 injection is the blue line. Post-injection, we follow the red line. And it's easy to visually see that the red line is clearly different than the blue line. It's less. If you look to the chart on your right, this is called a Wilcoxon-Mann-Whitney test, and it's a non-parametric test. And I'd like to just point out some characteristics. How many data points do we have here? We have 99 data points. And if you look at the mean pre-emplacement, which was 3,806 milligrams kilogram naphthalene, post-emplacement at 30 months, you're at 2148. You can also look at the median if you like. The mean is, as you recall, is the value, all the values divided by the number, right? Where the median is the actual middle of the data. You go from 1550 to 1010. Are these two lines different? Well, the p-value is 0.001. We'll just call it 0.002. So 2 in 1,000 chance that these two lines are the same. This is a similar graph for ethyl benzene. In fact, it's amazing how similar they look. When I was putting this together, I thought, oh, I made a mistake. I put naphthalene on both of them, then looked at them more carefully and realized no, I didn't do that. But you see also in the ethyl benzene at 30 months, you see the mean is lower, the median is lower. I didn't point it out on the other one, I should have. Even the standard deviation is lower. And your p-value is very similar, very small. The likelihood that the two lines are actually the same by chance is quite small. So if I looked at all the data and walked you through each of those graphs, what would I find, or what would I be able to show you? That the median difference before and after BOS 200 injection from soil samples from large data sets, benzene, it's 3.6-fold lower. Your half-life is about a half a year. In toluene, it's 1.5-fold lower. Your half-life is 1.16 years, and you can follow the data through. Now, the next question you're going to ask is, well, that's good, but how do I know that that's any different than what I would have expected had you just not done anything? Maybe this is what the half-life is in the kind of soils that you are looking at. So I don't have a lot of other soil data from sites where we didn't do anything, but I do have water data from sites that were left basically unremediated for many years. And where this came from is the underground storage tank program in Kentucky. There was a time when it ran out of funds, and for the most part, very little work was done. And an individual in Kentucky who works for the state, and I worked for the state at that time, we went through and looked at that data. The other individual, by the way, is Larry Hughes. And we looked at that data and put together a set of rules that essentially said, well, yes, they could have excavated the tank, or they could have even tried some remedy, but there was basically no remedy in place, and nothing had been done on the site for many years. I really don't remember what the number of years was now, but it was a good number of years, such that we thought that the site, whatever it was doing, was naturally remediating. And we looked at the monitoring well data that had been collected and was in the state database. And we found that for benzene, the half-life was about 3.2 years as a mean, or 38 months. I put together BTEX as a whole because this was for a presentation, and I needed to kind of move through the data quick and couldn't move through each of the individual contaminants. But for BTEX as a whole, we are looking at more or less 4 years. This particular data is from the bluegrass region of Kentucky, which would have been where the soil samples that I just previously showed you came from. Now, this is a California study. It was done by Tom McHugh. Some of you might know Tom, maybe not, but the maximum concentration rate per year, I converted the rate to a half-life. And so for benzene, you see the half-life is 3.8 years from this California data. And this is a published paper, and the way they did this is very similar to what Larry Hughes and I did, was going into the database, looking at sites, pulling together a large group of sites, which we thought we could look at half-lives from because they were not perturbed by effort, and collect these kinds of numbers. So the California number at 3.8 years, the state of Kentucky number at 3.2 years, all from groundwater, are probably pretty reasonable ranges for half-lives, at least on benzene. Now, it would have been nice if I had soil data, but I didn't. Nevertheless, you would expect the soil data to have a much shorter half-life than the water data. At least that's what I would expect, though the two clearly are linked, right? Okay, so the half-life in soil cores post-BOS 200 emplacement. Our half-life was a half year. What we've been able to estimate from, okay, someone says I am too loud and I need to decrease my sound. I have no idea how to do that, so I'll talk a little quieter, if that makes any sense. So using the Kentucky estimated groundwater data, we had 3.2 years. The California was 3.8. Clearly an improvement against those soil samples. Now, a question that'll arise also is, well, carbon isotopes, that seems to be very definitive data. Do you have carbon isotope data? And this comes from aquifer material from a Tennessee site, which as far as soil types, it would be near Cookeville, Tennessee. It's not going to be a whole lot different than the bluegrass of Kentucky. And what we see is the lower part, minus 29 down to minus 32, that was pre-injection. The other is post-injection. And what you look for is the gap between. And the gap between is over 2% per thousand, which, at least if you followed EPA guidance, older guidance, they would say that this is, proof of degradation. I would say it's a strong indication of degradation. It would be nice to have other backup data, but in some sense we do, right? We've shown you some soil, now we're showing you some carbon isotopes. This is similar data across a BOS 200 barrier. And what you see at 0 is up gradient. And as you move towards 50, you start going through the barrier at before 100, you're on the other side of the barrier. Obviously we don't have a 50-foot barrier, but you will notice that the enrichment increases over control for both ethylbenzene and xylene, and you see the same thing for propylbenzene. This is not as impressive as the other picture, but considering that you go through the barrier and then your numbers start to drop off on the other side of the barrier, that would be very consistent. So there's a lot of power in that pattern. So now we flip back over to the microbial populations, because what I've shown you so far is that microbes seem to grow on carbon, that we're able to remediate the soil itself, that we have been able to collect carbon isotope data, which demonstrates biodegradation. And now we're going to look at the microbial populations. And what we have on the left in orange is your unimpacted background. So before we actually took any remedial step, we collected these samples. And what you see is that total bacteria is about 2 to the 9th. Sulfate reducing bacteria, we'll call it 4 to the 7th. Iron reducing bacteria, about 5 to the 5th. If you look in impacted area prior to injection, you'll see that the numbers are generally higher. So it's 10-fold higher for the impacted over the unimpacted total bacteria. The sulfate reducing bacteria are, 10 to the 9th, right? Versus the iron reducing bacteria, which are also up quite a bit. 1 month post-emplacement, we seem to be stable or possibly with the iron-reducing bacteria a little bit lower. But look at 5 months post-emplacement. You know, we're clearly tenfold higher than the impacted area had been before. In most cases, the iron reducing is kind of holding its own. And you can see that on the chart with gene copies per liter over time. The initial count is in blue. The post-injection count is in orange. So in response to the BOS 200 injection, we are seeing higher microbial counts. What about functional genes? So this is a big list of functional genes that Microbial Insights can do testing for. Now understand, they're just testing for the gene presence in the DNA, okay? So you have both aerobic biodegradation genes and anaerobic biodegradation genes. So as an example, you have benzyl succinate synthase, which carries out this reaction, reacting fumarate with toluene to give you benzyl succinate, and then that goes on to be further metabolized. So it's an example of how that a functional gene out of this list might work. So here are several functional genes in aromatic metabolism. So you have your unimpacted background, your impacted, 1 month and 5 months. And you can see that you really don't have anything for this actually toluene dioxygenase, but we're not going to go through each of these. I'll spare you that for the moment. But you'll see that essentially you really had very little and you got some increase. And that occurs across the chart, which I've illustrated below in a diagram. This is the impacted background, 1 month, 5 months. Impacted background, 1 month, 5 months. So you can essentially follow the green balls across and see that post-BOS injection, you actually have increased aromatic metabolism. Aerobic aphilic metabolism. So this is your linear hydrocarbons. You see a very similar pattern. You look at the green balls versus the impacted background and you see an increase. You don't always get an increase with every microbe that would carry a particular gene, but you get an increase with most of them. And then here is the anaerobic metabolism. And anaerobic metabolism, of course, is what is associated most with activated carbon injection. But at this point, we're only at 5 months post-emplacement, right? And so you would expect over time to see these increase even more so than they are. But you start with very little in your unimpacted and impacted, and you increase 100-fold. So next generation sequencing. This is, I think, some pretty interesting data. And what I'm showing you right now happens to be the control data. So you go to the site, pull samples, you sequence the DNA, and you look and say, what microbes are present on this site? And you'll see of these 3 samples, they all tend to be rich in Smithella, very well-known alkene degrader. And we could go through a lot of these others, but I'm still kind of thinking about all of this data and how I want to put it together and present it. So I'm really not quite ready for that. But if you look in the middle column, and I'll just walk through that, you see Smithella, you see Methanotrex solingini, Dihalocoides McCartei, which is kind of surprising, right? There may be some halogens on this site, but you can also see Dihalocoides on sites that don't have chlorinated or halogenated compounds. Pseudomonas argilosa, a very well-known aromatic degrader. There's another Pseudomonas after that, also an aromatic degrader. And so you see, really, even before you've done anything on the site, you do have some sense that biodegradation is occurring. This particular population is probably not very dynamic. It's certainly not rich in various microbes. It tends to be very narrow on Smithella, which means that Smithella doesn't do aromatics and it does a limited range of alkenes. So that's really what the site is working on at this point, pre-injection. Again, it is degrading. It's just not very dynamic. This is post-BOS 200 plus injection. Now, notice most of what I've been pointing at to this point in time have been BOS 200 injections, and that's because we have the most historic data for that. But BOS 200 plus, we've been collecting a lot more microbial data. And I think the one thing I would like you to see immediately is look, it hits you in the eye, the number of microbes that you start seeing. Instead of being, half a dozen microbes making up 100% of the population, I had to stop at 1% in the presentation here because there's a whole bunch of microbes below that. And you'll see, so immediately, this is a richer population. There are a lot more. Now, I have placed a little Pac-Man to indicate microbes that degrade hydrocarbons, be they alkenes, alkenes, or aromatics, and shaking hands to indicate microbes that support degradation in some manner. Actually, the two charts on your extreme left and extreme right are not completed. The only one I've completed is the one in the middle. Again, still thinking about how I want to present all this, how I want to weave this data together, and I'd like to see a lot more before I do so. Nevertheless, post-injection, you got geobacters. You see a number of geobacter, which is very interesting because this is probably doing, extracellular electron transport. You see a number of Pseudomonas, and in general, a much richer, broader population. And that's, I think, the easiest way to present as much data as I have in front of you is to look at some of that taxonomic diversity. So we started off, and this would be the top line, the CHO1 abundance index. We started off with a CHO measurement of 3 in blue, which means that for our control, we didn't have a lot of unique individuals, and that's what the CHO measures. It measures the number of individuals present, singles or doubles. In other words, your minority populations, what do they look like in the microbes? And the reason that's important is a lot of times you can think that a microbe is not present in the subsurface. If you change environmental processes that are going on there, you might have a release of a contaminant, you might add a food source, you might get a seasonal change. Those that you didn't see before come up and start playing an important role. Then maybe the season changes and they go back down, or the contaminant is degraded and they go back down, or you get a degradation product, right? So you actually need to see those minority players deep in the population to know, in a sense, you have a deep bench on this team. So you go from your control to your impacted control, you start seeing more of those low number players. But post BOS 200 plus injection, you go to 24. This means there's a large number of individuals, in a sense, that are waiting there on the bench to play. The Simpson Diversity Index tells you something about evenness. And what is meant by that is, are you sitting on all Smithella, right? Are you at 75% Smithella? Well, that's not very even, is it? Most of the time, microbes perform best when they have lots of other microbes around them, because if you're a microbe, you're very, very small, and you can't really perform all the metabolic functions you might like to. And it would be good to farm out some of those metabolic functions to other microbes around you. So the healthiest populations live in consortia, and consortia tend to be more even. So evenness is important. And we look and we see that we get an increasing evenness post BOS 200 emplacement. The Shannon-Weaver index tells you about the total number of everybody, the number of species. And it's just the natural log of the number of species. So you see that the number, how many people are there, the abundance is increasing post-injection. So this is a good way to look at that previous data without actually getting into the metabolic details. Now, one thing you might wonder is, well, you pointed out this BOS 200 plus. How is that really different from BOS 200? So I thought I would show you that. This is a BOS 200 only site. This happens to be at 3 years because we have that data. This is what the site, on the left is what the site looked like prior to injection. On the right is what the site looked like at 3 years. Now keep in mind, for example, if you look at Smithella, a known degrader, you have Smithella on both sides. But remember that on the right side, you're tenfold more abundant. You have more individuals to begin with. So the Smithella concentration on the right is actually much higher than it is on the left. But yet you also have a much richer population. You see the Geobacter and the Geothrex dropping in. You also see the Dichloromonas, which again is kind of interesting. You have it on both sides still. So a richer population. And a richer population is a population that has more metabolic options. It's a population that is healthier. This is 200 plus on the left, which I just popped up, compared to BOS 100. And again, this population, the BOS 200 on the left, is yet more abundant and more rich. You notice that on the right, Extensomonas down to Geothrex, that basically covers up close to 100% of your population, right? On the left, you go to from Acetovorax, a very good hydrocarbon degrader, by the way, all the way down to, well, happens to be another Acetovorax soli. So you're getting a greater richness, right? And I haven't finished this chart out completely so I just marked off a few of the hydrocarbon degraders that struck me as I looked through it, but you have a good healthy population. Now, another question that might arise is, okay, yeah, you're injecting carbon in these hydrocarbon sites and you're getting these rich populations. Maybe no matter what you did, if you injected something in the ground in a site, you would get a similarly rich population such that this is not unique to hydrocarbons in any way. Well, this happens to be the pattern for CAT 100. Now, CAT 100 is designed for chlorinated. So you have a different contaminant, and the carbon is modified by having an iron molecule integrated into the carbon, not mixed with, but integrated. So you have a different scenario. Look at your control on the left. What you see is Methylocystis, Methylobacter, Methylocystis rosa. Essentially, you have a population that prior to injection is doing some degradation of the chlorinated, See, you can see the dihalocoides spectrum down near the bottom of the list. So yeah, you're probably getting some, and you got Pseudomonas aeruginosa, you're probably getting a little bit of degradation. But again, it's not dynamic, it's not rich. They are living on methanol and other post-fermentation products, not too good. Look on the right. This is post-cat injection, several years actually. And notice that you're maintaining a rich population of Dihalocoides macari at 80%. I shouldn't use the word rich, but I guess it's rich for Dihalocoides, right? It's 80%. So this is a different bias. You're not seeing the, what I should have used is the broad abundance and richness that you were seeing in the hydrocarbon. You're kind of focusing a lot on the Dihalocoides. Now you have a number of Dihalobacter below that are important. You have plenty of supporting bacteria. A number of them produce hydrogen, which is necessary to keep Dihalocoides macari moving forward. So it is good that you have those other supporting microbes, and they are there. But you can also look at this and see that this is fundamentally different than you saw for the hydrocarbon sites. So we'll stop there, and I'll kind of walk back through it, and then we'll have some questions. I apologize for the technical blurps here and there, but hopefully you enjoyed them. Activated carbon provides a space in which microbes can grow, and when they grow in that space, they build a biofilm and they build a community, and that community, when supported with the right materials, such as we have in BOS 200 plus, will be a community that is rich, that is abundant, that sustains a number of minority players, which can increase or decrease as the environment changes, and that is sustained over time for years, in fact, and that is not just, oh, I stimulated this aquifer by putting something in it. You can see it's very different than what you would expect to see from a chlorinated site with a different carbon and a different stimulant. So what kind of questions do we have, or what kind of things did I just bobble? What do we got, guys?
All right. Thanks, Ed. So this is Mike again. Again, feel free to, any questions you have, go ahead and write those in. Just a couple questions to get us started here, Ed, that came in. One question was about the CSIA and sort of what would be a recommended, if you're going to deploy this out. There's actually a question on the microbial diagnostics, kind of similar, as you're going to, if you're looking to deploy those techniques out on a UST site, what would be your recommendation as to, from a baseline standpoint, number of samples, like frequency, that sort of thing?
Okay. Yeah, this is an interesting question. And that even the data I showed you was not what I would like to have had. It was collected by a state agency, and they gave it to us, and we greatly appreciate it. And it's also true that the standard by which you judge success for carbon isotopes has changed, say, from what it was even 10 years ago. Now, that doesn't answer your question. It depends on what you want to look at, and what most people are interested in is benzene. And benzene, in some sense, is hard to catch because you really need to degrade about half the benzene in the area in which you want to sample, so that you will have enough carbon isotopes to discern the difference between background and degradation, right? So you really need to, one, look very carefully at your monitoring data. and you have to be prepared to get out to the site at the right time. You can't just say, well, we'll show up every year, and one year we'll just decide to collect carbon isotopes. You probably won't be successful in doing that. The other thing that's important, which I don't have in my own presentation here, is you should collect hydrogen isotopes also. It is possible that you can look at carbon isotopes and basically spend a lot of money and just not be able to discern a difference, even though there really is a real difference. And it has a lot to do with just trying to sample the aquifer. It has to do with laboratory processing and other things. So the first thing is you want to look at about, try to sample around the time when it's about half degraded as far as benzene goes. Also collect carbon isotopes and make sure that you have good controls. In other words, you have to have an area that you didn't, if you're looking at treatment, that you didn't treat, right? Now, you can do that one of two ways. You can do that by looking outside the area, or you can do it with the flow, right? Up gradient versus down gradient. I don't know if that was a great answer, but there you go.
No, I think I think that was great, and I guess it would be a similar answer for from the microbial diagnostic standpoint too, correct?
The microbial diagnostics is a little bit harder because you have different ways of looking at it. Okay, so the one set of data that I showed you, the microbial insights data, and let me flip to it, unless I'm headed in the wrong direction. I am not, and here we go. This data is only going to allow you to see what the test actually tests for. Okay, so if you look for toluene dioxygenase, which is the very first one, the TDO that you see on this list, if a microbe carries that gene and is present on that site, it will come up in your cell counts per mil on this kind of chart. But what if toluene dioxygenase is not very important on your site? Well, it may not come up on this chart. And so while this chart is very extensive, and yeah, I mean, they've done a beautiful job. I'm not picking on their process at all. These things are great. They're very simple to understand, and they're definitive for what they show you. But what they don't show you is that there are a lot of other microbes that are very important in sustaining the health of your degraders. Okay, so let's say you have a number of microbes that produce acetate, which is degraded by the degrading microbes. You are going to be able to work on those hydrocarbons to a much lower concentration in that aquifer and keep that microbial population richer and healthier for a longer period of time if they have those supporting microbes that in a sense supplement the diet, right? A diet of pure hydrocarbons is kind of tough. Not only is it kind of tough, it's also inconsistent. You don't always have them when you want them. So that's one thing. There's also electron cycling. Some of these microbes will take nitrogen and just keep recycling it. And you want to have those to support your degraders. You'll never see it in this QuantArray analysis, but the QuantArray analysis is very easy to present. It's very easy to understand. And when would you collect it? Well, that depends on exactly what product you are using and something having to do with your geology. If let's say, for example, you just injected a bunch of microbes, okay, so you were doing a bio augmentation, well, you wouldn't want to turn around and use the test right away, right? You would want to see if that bio augmentation were any good 9 months from now or a year from now. So most of what I showed you for our site, this is early, what you see on your screen right now, and that's what the contractor on this site wanted to do. When this will become most meaningful is when you can demonstrate that these microbes are functioning 2 and 3 years out. And that's why I walked you from the QuantArray data to the next generation sequencing. Now, no one asked the question, but I'm just going to blab here a minute. The next generation sequencing shows you everything that is in the population. The difficulty is then you have to go in and interpret it, where the next gen sequencing interprets it for you. So one of them is broad, very definitive. It tells you a lot of information. That would be the next gen sequencing. The Microbial Insights gives you a very easy to understand, are the degraders here or not kind of answer.
Okay. Here's just a simple one. Can you just briefly describe the difference between BOS 200 and 200 plus?
Yeah, probably the main characteristic is BOS 200 plus is supported by starch and some other nutrients. And starch is not terribly soluble, so once you put it into the aquifer, you have that supplemental food source that lays in the aquifer and slowly dissolves. And what this does, very similar to what we were talking about earlier, living on hydrocarbons, for example, or chlorinated compounds or any other contaminant is a hard way to make a living for microbes. And so, it's kind of like, inviting a bunch of invalids to, become your football team, right? You really don't want to do that. What you want to do is you want to find, healthy, strong people who are well fed for your football team. And that's what you're trying to do with the BOS 200 plus. We've looked at it and we've realized, if we supplement, we can build a much richer population. And so let me give you an example. A lot of people know about geobacter and the value of geobacter in allowing one to transfer electrons without having to build intermediate chemicals. It's a much faster way to degrade an item is to be able to transfer that electron directly as opposed through intermediates. You won't see as many or as rich a population of geobacter in the old BOS as you do in the BOS 200 plus, simply because that extra support makes a big difference. It does the same thing too when you're headed, you end up keeping the population in fermentation a lot longer and you build a richer population so that when you move into methagenesis, which by the way, some people think is a really bad thing. It's not a bad thing. Methagenesis is a really good thing because ultimately you want to break your contaminants all the way down to the basic molecules, right? And the only way to get there is through methagenesis. But what you see with the BOS 200 plus is you hit methagenesis and you get a really quick steep climb and then all of a sudden it just plateaus. And I'm seeing it in the microbes. I don't have it on a chart yet. Can't really show you. But I think what's happening is the other microbes are kicking in and using that methane so that our methane plateaus out, which is great because you're getting both the advantage of methagenesis and you're avoiding the disadvantage. And that's similar in electron transport and other aspects of what's going on in the BOS 200 plus.
Okay. Well, we've got about 5 more minutes. We'll try and get through a couple more of these. The question is, any thoughts, plans to add biofilm attributes to your analysis?
Yeah, I would like to do some biofilm work. I think it's clearly important. I've already started doing some work at the University of Kentucky, confocal work, trying to get some good look at the biofilm and also what microbes are there. So we're in the beginning stages of that. But clearly, one of the things I've tried to emphasize is the BOS 200 plus is giving us a solid, sustained population over time that is degrading not just for a few months, but we hope that, like the BOS 200, that it'll go on for years. And key to that is being able to build a dynamic biofilm community, and to be able to show that to people, to me, would be really exciting. Now, we already can show you from lab samples. I can show you all the biofilm you want from lab samples. Lots of publications in that also. But I'm talking about field samples. That's what will be exciting.
Excellent. Next question, do you have any data comparing activated carbon to biochar? Is biochar more effective at giving the microorganisms growth medium?
Yeah. There's a lot of publications on biochar, tremendous, and a lot of them come from China. And the reason being is biochar is very cheap. It's very inexpensive, and cheap sounds like it's a bad thing. It's not a bad thing. It's inexpensive, and expensive would be good. But the absorption capacity of biochar relative to activated carbon is poor. And its value as a growth medium for microbes is also rather weak. So, there have been a lot of studies trying to make up for the difference by using more of it, supplementing it, etc. But it just doesn't work as well. And one of the reasons it doesn't work as well is the porosity of biochar. It tends to be, if you could look at it under an electron microscope, it tends to be more like a net with big holes in it. You don't have near the number of micropores that you do in activated carbon, coal-based activated carbon. And what you'll see is that's where the absorptive capacity of the carbon is. And the absorptive capacity is important because in a sense, you concentrate your food source. You make a place, a pantry for the microbes to be near. And biochar just doesn't pull that off to the same degree. And it's unfortunate because it would be really great. The cost of biochar is really cheap. And for countries that are not as wealthy and have pollution problems, you can understand why they're interested in biochar, and there have been a lot of publications on it. But it's not as good as activated carbon. Sorry.
Ed, thank you so much. We're up against the hour. So there were a couple questions that we didn't get a chance to respond to. We'll go ahead and reach out to you directly. I see most of you have hung around for the full hour, so thank you. Thank you so much. And again, if you have any questions, reach out to Ed or myself. And again, thank you for joining us today. Take care.
Okay, thank you.